Hey everybody. This podcast is going to deal with the next step in our look at enzymes, which is that very, very, very important group of proteins that do so many things for us and all of life on Earth. There are many things that can affect the ability of an enzyme to do its job, to catalyze its chemical reaction that it's in charge of, to make that reaction go faster or slower, different environmental conditions. Okay, so what we're going to look at is what those different things are that can affect enzyme function and exactly how they do so. This is a rundown of the list of the things that we are going to look at. Okay, so when we're done, you should have some understanding of how all of these things affect enzyme function, and there are some things that some common threads you'll see as we go through. Now, the first thing that can affect how fast an enzyme can do its job that it is supposed to do is how much enzyme you have, how concentrated is your enzyme in your among the substrate molecules that you are trying to react, that you are trying to make undergo a chemical reaction. Okay? Now, with each of these, we're going to look at a graph, much like the one that we create with toothpick ace in class. That graph is showing us on this side, on the y-axis, the reaction rate, and on this particular axis, this is the variable, our independent variable that we're looking at, enzyme concentration. So if you think of this as a little experiment, let's say we are going to manipulate how much enzyme we put in with the substrate we want to cause to react, and what we're going to measure is how fast the reaction goes. And what you can see is as enzyme concentration goes up, if we've got a cup of some substrate, okay, let's talk about this in terms of hydrogen peroxide, for example, because that's the chemical we'll work with in our lab. In our lab that we're going to do in class, hydrogen peroxide is our substrate. It is the thing we want to react. We want to actually break this apart into oxygen gas and water, two harmless substances. The enzyme that does this is called catalase. Okay, so let's say this is a bunch of hydrogen peroxide. So what this graph is showing is that the more catalase molecules I add to the hydrogen peroxide, the more I add, the faster my reaction goes up to a point. And as you can see, there's a point where I can add more and more and more and more enzyme, but the reaction is really not getting any faster. In fact, it's just kind of leveling off. It's going as fast as it is possible to go. So let's look at what would explain this phenomenon. I want you to think of the substrate, the hydrogen peroxide, if you will, the chemical that we want to break apart, as a door, and the enzyme is a key. Okay, now if I've got a bunch of doors, for example, here around school I got a bunch of doors, and I have a single key. I can open all those doors with that single key, but it's going to take me a while to do all of them. However, if I increase the amount of keys or people with keys, then I could have one person with a key opening this door, one person opening that door, another person opening that door, and so on. And so I could essentially open all of them at the same time, because I would have a bunch of keys to do. I wouldn't have to do one door at a time with one key. That's kind of how the enzyme concentration affects the chemical reaction. The more enzymes I have, I can break down that many more hydrogen peroxide molecules at once. The thing is, beyond a certain point, it's not going to make it go any faster. I could add even more keys to my scenario right here, but I have 5 doors, and even more keys more keys than there are doors is not going to make those 5 doors get open any quicker. And each door can accommodate only one key at a time, so it just doesn't matter. There's a point where adding more keys just doesn't help me go any faster. Same with the enzyme. Adding more enzyme doesn't help after a point. So generally speaking, as enzyme concentration goes up, the reaction rate speeds up as well. The more enzymes, the more times they are bumping into the substrate and reacting with them. But the reaction rate levels off because eventually the substrate becomes a limiting factor. There's plenty of enzyme, but not all of it can find a substrate to react with. This is as fast as we're going to go. Okay, so next, we can look at not enzyme concentration, but substrate concentration. How much substrate do I have? And we see a similar thing. Here's my y-axis with what I'm measuring, how fast my reaction goes, my reaction rate, and this is showing how much substrate. And as substrate goes up, let's see what happens to the reaction rate. Sorry. There we go. As the substrate concentration goes up, just like with enzyme concentration, I get a quickening of the reaction rate initially, but at some point it just can't go any faster. It's going as fast as it goes, and the rate levels off. Okay, what's happening here is that as we saw in the graph, as the substrate goes up in concentration, the reaction rate speeds up because, again, the more substrate I have, the more often it's going to bump into enzymes, and they're all going to be reacting and going crazy. But once all of my enzymes, which are being indicated here in the red, once they all have, each of them has a reactant in its active site, I don't care if I keep adding more substrate, more reactant. If I keep adding more, they're all occupied and they're going as fast as they can go. So continually adding more than those enzymes can hold doesn't really help me increase my reaction rate. This is as fast as it's going to go. Okay, the enzyme is what we call saturated. Okay, it's handling all the substrate it can handle. It will continue to handle these additional molecules as it breaks down this. It'll take another one, but it's not going to go any faster. Every single enzyme is going as fast as it can go. Okay, so temperature can certainly affect reaction rate. In this case, what we're seeing is that if we go from a low temperature and temperature increases this way, we see that the reaction rate is very low in the cold range, peaks out here in the middle for a particular enzyme, and then again gets very low as we increase it, increase the temperature higher than that peak. This is fairly typical for lots and lots of enzymes. There is, for most enzymes, an optimal temperature, meaning a one best temperature at which they work. It's the temperature where, remember, part of what makes enzymes work is that they bump into their substrate. They bump into the thing they are supposed to react with. And so you want to have it a certain temperature, a certain amount of heat to ensure that enough molecular collisions happen. There's enough kinetic energy that they're actually going to collide and bump into each other. But obviously you don't want the temperature to be so hot that you begin to make the molecules of the enzyme move so quickly that the enzyme itself begins to lose its shape and begins to fall apart or become denatured, as we've talked about. So for human enzymes, they seem by and large to be adapted to work best at a temperature somewhere around 35 to 40 degrees Celsius. Human body temperature is 37 degrees Celsius, so it makes sense that the enzymes that we have in our bodies would be selected to work at a temperature at which our human body normally exists. So if we increase the heat beyond that optimal temperature, then we risk denaturing the enzyme, disrupting the bonds in the enzyme and between the enzyme and the substrate, and just basically making the whole complex fall apart. Remember, a lot of these bonds are fairly weak in the first place, so they're going to be the first to go. Hydrogen bonds would be among the first to be disrupted by a temperature increase. So you should know this word. We've heard it before. Denaturation, or the denaturing of an enzyme, is when it loses its very important shape. The active site is no longer shaped properly. It can't fit its molecule. It's not going to work anymore. Okay? Now, if you decrease the temperature below the optimum, so remember, here's my optimum. We know what happens if I increase the temperature above the optimum. I'm going to risk the denaturing process. But if I lower it below the optimum, I'm not so much risking misshaping my protein anymore. It's not necessarily going to fall apart like it would when it's hot. But what cold does do is it reduces the number of collisions, because we're going to lower the kinetic energy of the molecules and they're not going to bump into each other as often. So I'm going to make my molecules move slower and therefore decrease the number of possible collisions between the enzyme and the substrate, and then that is going to lower my reaction rate. So what we see here is the reason why this graph looks the way it does. You chill it down, they're not going to bump into each other, the molecules don't bump into each other as much, and so our reaction rate is decreasing. You heat it up and the enzymes begin to denature, and that's why we see the rapid drop off with a temperature increase. This 37 degrees is the optimal temperature for many human enzymes. Okay? Now, there are different enzymes in different organisms that have different optimal temperatures. Okay, in a human, the optimal temperature for most enzymes is 37 degrees, but there are bacteria that live in places like this geyser, for example, whose optimal temperature is going to be tremendously higher. 70 degrees Celsius, 100 is boiling. So these enzymes and these organisms are going to be adapted to tolerate a much higher optimal temperature. Which makes sense. Excuse me, I think I just burped in your ear. Okay, pH can also dramatically affect how well an enzyme functions. What we see here is, again, the normal condition for human enzymes. Most human enzymes, since our normal internal pH is somewhere around 7, it's not 7 exactly, most human enzymes are going to be adapted for that pH level. However, even within our own bodies, there are some enzymes that are adapted to work best in a dramatically lower optimal pH. Pepsin is one of those. Pepsin is a digestive enzyme found in the stomach, and if you'll recall from perhaps past learning, in your stomach there is hydrochloric acid and lots of it. If pepsin was adapted for a 7 pH, it wouldn't last a minute in there with all of that acid in the stomach. So pepsin is adapted to a much lower and acidic pH, which makes sense. There are other enzymes in your digestive tract. Trypsin is one, and trypsin is actually secreted from the pancreas into the small intestine. This is the stomach, this is the small intestine, and when the partially digested food exits the stomach into here, basic substances are added to this acid to try to neutralize it. So the conditions in the small intestine are quite different from conditions in the stomach. And in fact, you'll see a more basic solution in here, a higher pH solution than you would see in the stomach or even in the rest of your body. And so trypsin is actually adapted to survive well, or survive. Please remember, enzymes are not living things. They are molecules, but to not be denatured in a higher pH than 7. Okay? pH affects enzymes because if you'll recall when we talked about homeostasis and we had our pH discussion, a solution, here's a beaker, it's got an acid in it. What that means is it's got a bunch of excess hydrogen ions in it. And these hydrogen ions are very, very reactive. So if you dropped an enzyme in this solution, these hydrogen ions would want to react with it. So changes in pH can cause these hydrogen ions to react with the enzyme, or if we drop that enzyme in a basic substance, for example, that's full of hydroxides, we've dropped the enzyme in here, and this might rob, these hydroxides might actually rob H's hydrogens from the enzyme that we put in there. Either way, you're going to be dramatically changing the shape of the enzyme. So it's disrupting chemical bonds within the enzyme and disrupting the three-dimensional or conformational shape. Okay? And this is just detailing all those things that could be affected. Secondary and tertiary structure, just basically denaturing the protein. So, again, this is just refocusing on what we just talked about. Optimal pH, most human enzymes is 6 to 8, but it depends on where the enzyme is. Okay? Alright, salinity is just how much salt you have in your cells, in your body. And so again, we kind of see a similar path. There is an optimal salt concentration above which enzymes don't work well and below which enzymes don't work well. So salt concentration is basically kind of similar to pH. If you have salt, remember salts are made of ions, a sodium ion and a chlorine ion. They are held together by their opposite electrical charges. So if you put a salt in a solution, you're going to have cations and anions floating around in there that are rather reactive, and much like the hydrogen ions and hydroxide ions, they will react with the atoms in your enzyme and disrupt bonds. So again, it's just a matter of these chemicals reacting too much or too little with a particular enzyme, and again affecting its shape. Okay. Enzymes are generally not going to be tolerant of extremely salty conditions, and the Dead Sea, this note is dead for a reason. There's not much life in there because most enzymes just can't tolerate the salty conditions, and so if you can't have those enzymes, you can't have life. Alright, very quickly, I just want to touch on some things we've talked about. Actually, no, I think I'll stop right here and we'll pick up on this a little bit later. So that's it. Thanks a lot for watching. See you in class.